I think the challenges in our area is that the risk we're looking at is so extremely rare. If you look at a compound that we believe is proarrhythmic, let's say Sucipride that was on the market until some years ago, the incidence of severe proarrhythmias were still in the range of 1 in 100,000. That means that effectively you need to study almost 1/3 of a million patients to capture one event. So that is, of course, a major challenge. We can't do this in normally sized clinical trials. We need to come up with more intelligent solutions.
In clinical, I think there's two phases there. Obviously, if we can develop a biomarker that's applicable to healthy volunteer studies, is one aspect, and then the other aspect would be in patient studies whether we can have biomarkers that would be an early predictor of efficacy in a patient population.
So this is a very important arena to work on, and what happens is you need a prospective study that is long enough to have a very good predictive biomarker, and during this whole development we need industry, academic, clinicians, biostatisticians. They need to be involved during the whole process of it. The challenge is that so far the funding and the interest and the aims of these different people that need to be working together has not been met. The funding only lasts for 1 or 2 years. Either the industry only is interested in the latest development and is not interested in the earlier development. Clinicians and scientists need to work together for a longer period of time. So all of that has to come together during enough time to make sure that your predictive biomarker is well developed and is validated.
Well, one of the challenges is that we have an opportunity actually with a lot of new technologies coming to bear, imaging and things we hadn't even heard of 10 years ago. But a big challenge is the reproducibility of these techniques and making sure that they are kind of validated. So there's both external and internal validity. The internal validity is that you have a robust test with the biomarker, and the external validity is, does it really translate or predict from the animal model to the humans?